The overall goal of the following experiment is to measure RNA polymerase II transcriptional dynamics in living cells as it transcribes from a specific gene in real time. In order to follow transcription on a specific gene sequence, a gene construct is stably integrated into a human cell line. This gene enters in several copies into one random site in the genome and forms a gene locus containing a tandem array of this specific gene. When gene expression is activated, RNA polymerase II is recruited to the gene and moves downstream along the DNA strand, generating the mRNA molecule. In order to tag and detect the transcribed mRNA, a series of unique sequences, or MS2 repeats, that serve as binding sites for a fluorescent protein, YFP-MS2, have been fused into the gene sequence. The rate at which the MS2 stem loops are formed corresponds to the rate of transcription. Fluorescence recovery after photobleaching, or FRAP, is then used to visually detect ongoing transcription. A strong laser beam is directed at the active transcription site to photobleach the YFP-MS2 molecules, and the recovery of fluorescence is followed. The rate at which the fluorescent signal recovers reflects the rate at which RNA polymerase II is transcribing. A recovery curve is obtained that shows the measurement of the increase in YFP-MS2 intensity at the transcription site over time. The transcriptional kinetics can then be extracted from these data. The results obtained show that RNA polymerase II elongation rates in vivo can be measured, and that these are much faster than previous in vitro measurements.